Okay, so my name is José Mombach. I'm professor at the postgraduate course in physics of UFSM, and I'm going to talk about the laboratory of complex systems, which I am uh, the responsible person. So the three main areas of research in my lab are superrepellence, biological tissues modeling, and gene network dynamics. Superrepellence is a very interesting phenomenon, which was first observed by a German biologist. He observed that the leaves of a plant called lotus, they were always clean, and this plant inhabits swamps, which are very dirty places. And he noticed that during rain, the plants didn't get, the leaves didn't get dirt. So he concluded that what was happening is that the water droplets sliding on the leaves of this plant would carry away the dirt. And for example, what you see here is that these leaves of the plant, they have small, is rugged, has these small structures, and when the droplet of water did not attach to the surface, and when they slide, they carry out the dirt. This is different from what happens when you have a smooth surface. When a droplet of water slides, it can't carry out completely the dirt. So that was called then hydrophobicity or superrepellence, a property that makes surface to become clean. And on this type of surface, what happens is that a droplet of water becomes almost spherical. They have a large angle of curvature, what you see here, usually above this value, 150 degrees. The surface is considered superrepellent. Then people was interested in this phenomenon. By the 70s and 80s, a lot of research started on the field, and people started to produce artificial surface like this one that you see here. This is an artificial structure constructed by generating pillars on a surface, and on these pillars, a droplet of water becomes almost spherical with large angles of contact. And there is two states of wetting, which is how we call that phenomenon. The wetting is when the droplet is able to penetrate in between the pillars or remain only on the surface of the pillars. When it's on the surface of the pillars is that this property of superrepellence appears. Here what you see is a classification of the behavior of a droplet on a surface. In the first case, we have the superhydrophobic state, which is a synonymous of superrepellence. The angle of the contact angle is large, about 150 degrees or more. In the second case, we have a hydrophobic surface when the angle is between 90 and 150 degrees. And the last case is a hydrophilic surface when the angle is lower than 90 degrees. And what we do in our lab is to simulate this behavior using computer simulations, okay? And in these computer simulations, we use a lattice, and we classify different regions of the lattice as being gas or liquid or a solid, is what you see here and in the previous figure, okay? This is an image of a real simulation, and this image is how we build it. It's just a small amplification of the simulation of the lattice. And here we have the equation that we use, the Hamiltonian used to define the energy of the simulation. Using these methods, we were able to explain some interesting phenomena in the field. For example, we were able to describe how the transition between these two states, the state in the case the droplet penetrate in between the pillars, and when it remains on the surface. Okay, and this is an important phenomenon use it to produce this kind of surfaces. So this is basically an outline of this first field of my research.
5 minutes, Mombach.
Okay, and the second area is the biological tissues modeling, where we have what you see here are two aggregates of biological cells. The cells are obtained from chicken embryos, so are cells from a chicken. And when they are grown in a culture medium, these aggregates are inside a culture medium, they attach, the cells attach and form these spheres that you see here, okay? And the properties of these tissues are important to understand how an embryo is formed. And also they are recently being used to produce artificial organic structures, okay? Here what you see is just two sets of aggregates in contact, and they fuse when they are put in contact. And it's similar to what happens when you have two droplets of oil on water when they fuse when put in contact. Why the cells attach? Well, what you see here is a cross-section of the membrane of a cell, and here is in these schematics is that cells have adhesion molecules on their surface, which makes them to attach to each other. And this property happens, that's why the aggregates become spherical, because they want to minimize this adhesion energy. And using these aggregates and a 3D printer that I show you in the next slide, the guys use this aggregate to build artificial structures like this ureter from kidneys, okay, to make transplants and so on. Using these aggregates, they can fuse and form a real 2D structure that can be implanted in a human being. And what you see here is one of these 3D printers that implant these small aggregates in a given structure using a sort of scaffold, using sugar, which is dissolved in water. They can form a structure that you want, okay? So this is one of the areas that I do research. And what you see here is our simulations published in this paper. What you see in this image is the real aggregates on top and the simulations just below, okay? And the next area is gene network dynamics, which we have done most of the research in the last years. What you do here is try to represent how proteins, what you see in this image, are different proteins that control if a cell is going to replicate, to die, to commit suicide, or to stop growing. And these are proteins that can activate other proteins. When you see the arrows, the pointing arrows means that they activate other proteins. And when you see this arrow with a bar, means that they inactivate the other protein. What we do here is to represent these protein interactions through Boolean algebra, representing here what you see in this box. These boxes represent the proteins. They have two states, 0 or 1, and they interact through Boolean operators or and not. In this way, we represent the functioning of the molecules inside the cell as a computer program, okay? And there are two approaches. One is on the left here is through differential equations, okay, which is the traditional way of representing chemical reactions. And what you do is in the right here, use Boolean. When we have hundreds of proteins, the mathematical approach becomes hard. Then we have to use the Boolean approach because it becomes much simpler and also is able to capture, despite its simplicity, is able to capture some important phenomena in these networks. And recent papers that we have published have gained some impact in the area. And this, the main, I would say 90% of the research in my lab is in this last field. Okay, so this is my contact. And this is my page in the ResearchGate social network. And if you want to have questions or want to make me questions about the subjects, I will be happy to answer them. Thank you.